Hello, students. Good morning, everybody. Today, let's start a new chapter in immunology called tumor immunology. See, after the year 1940, because of the discovery of antibiotics, the death because of the microbial diseases significantly decreased in Western countries. But then, the death because of the heart diseases increased, followed by cancer. And cancer is an altered shell. You know what really happens? In our body, we have trillion number of cells, and all these trillion number of cells, they definitely obey the signals given by the neighboring cells. But it so happens that um, in case of one day, among the trillion cell, one cell detaches from all other cells and it stops receiving the commands from the neighboring cell. Though the neighboring cell is telling that you stop cell division, the cell is not stopping, and then the other cell may tell that the cancer cell you die, but the cancer cell is not dying. So this actually results in the continuous proliferation of the cells. Um, anyway, and so this particular cell, it starts dividing and redividing. It forms a considerable size, and that's what we call as neoplasm. And in the cancers, we mainly say there are two types. One is the benign, where the cell is grow, it forms a tumor to a certain size, and then it stops dividing. These are called as benign. These are quite harmless. With the help of a surgery, one can remove the benign tumors. But then the second category is, so there are certain cells, so the tumor cells, they start invading into a surrounding tissue, and that's called as the malignant. That's what we say as a cancer. And sometimes it also happens that see some cells from the tumor, they detaches and they enter into lymphatic system, blood system, and they start proliferating even in the other organs. That's what we call as metastasis. What we say, the final stage of a cancer. Okay, now let's look at the difference between the normal cell and the cancerous cell. Usually the normal cell, I mean, we do have a homeostasis in our body. Okay, so for every type of cell there is a certain number. Once that particular number is reached, the cell growth is stopped. Okay, but that's what we say a normal one. It means that normal cells definitely responds to the density, but the cancer cells are not like that. Irrespective of the density, they keep growing. Okay, and then the normal cells, they proliferate only to certain generations. After that, they die. But the cancerous cells are immortal, they don't die. And in a normal cell, if you look at the length of the telomere, so the telomere is actually shortening as we grow. But in case of cancer cell, telomere size is not decreased, therefore they don't die. And as all we know that, so all the cells in our body, they keep dying because of apoptotic signals, but the cancerous cells, they don't die. So it means that they are overcoming apoptosis. Now, based on the origin of embryonic origin, cancers are divided into different types, of which, if these cancer cells, they originate from endoderm and ectoderm, we call them as carcinomas. In fact, 80% of the cancers in our body belongs to the class of carcinomas. And then, so if the cancer cell originate from the mesoderm, okay, then we say it's a sarcomas. Um, or if the cancer cells, if they originate from the hematopoietic system, then we call it as leukemias or lymphomas. And it so happens that, as I said, we do have a trillion number of cells in our body. It's not that all or many number of cells are turning into cancer. It's only one cell among the trillion cell which is transforming into the cancer cell. We have a proof for that. See, let's say if the person is suffering from leukemia, and if we take that blood sample and if we observe, we could see that in all the cancer cells there is a translocation of a part of chromosome from the 22nd chromosome. It is translocated to the ninth chromosome. Or you can say that in case of mammals, see in the females, we have two X chromosomes in the females, of which one we have received from father, one we have received from the mother. But in all the females, one of the X chromosome undergoes heterochromatinization. See, suppose if you observe the cancer cells, we could see that the same kind of heterochromatinization, okay, in all the tumor cells as it is depicted in this picture. You can see in all the tumor cells, I mean, you could see the same kind of thing. And now, remember one point, cancer is mainly because of accumulation of many number of mutations. For that, initiation is required. And suppose you want to study the cancer in the lab to induce the cancer, all that you have to do is you can take a mice and then you can apply a coal tar or tobacco smoke on the ear of the mice. Okay, if you keep repeatedly applying it, then so after some time, the mice they start suffering from cancer. I mean, these are called as the initiators. But once this initiator itself is not enough, we also require promoters. Suppose in that case, say there are certain chemicals called as tumor promoters. It may be porval esters, or say, suppose when we eat like chitrana puliyogre, where we put peanuts, and in the peanuts sometimes there will be bitter in taste, and that bitterness is because of the growth of fungi which produces aflatoxin, and these aflatoxins are actually they behave like promoters of cancer. So okay, and even the application of the lipsticks, this lipstick which mainly contains proestrogenic kind of compounds and heavy metals like lead, they may even promote the cancer. Okay, anyway. It's true that the cancer cells are multi-layered and they become round and they lose contact inhibition. Look at this picture. This is the cervical cancer. So the woman who is suffering from cervical cancer, they are subjected for Pap smear. Look at the first picture, that is A, where you have the all the cells. This is a normal uterine lining. I mean, you can see that the normal epithelial cells, they have a different shape. But look at the second picture and the third picture, the B and the C, where you can see that the cells have lost the shape. They are turning it around, and then there is increase in the size of the nucleus, and there is a change in the morphology of the nucleus. Then, um, say, with the cancer, probably because of the production of proteases, and these proteases would degrade. They act on E-cadherins and integrins, which are they have the intercellular matrix. Okay, or it may be even because of the defect in the enzymes which are involved in DNA repair mechanism, recombination, and replication. Okay, so for example, we get skin cancer called as xeroderma pigmentosum. In that disease is mainly because there is a defect in the DNA repair mechanism. Therefore, the patient, as soon as they are exposed to UV radiation, and we all know that UV actually induces mutation, but then the DNA cannot be repaired because of the defect in the DNA repair mechanism. That actually results in xeroderma pigmentosum. Mm. And the cancer cells, they have a high capacity of mutation. Therefore, because they are more subjected for mutations, it's very difficult to find a treatment for cancer cells. In fact, most of the cancer cells do have a double chromosome called as double minute chromosomes. And you know what really happens in that chromosome? There is a gene called as multiple drug resistance gene. This multiple drug resistance gene, which produces an enzyme which is actually bound to the plasma membrane, which produces ATPase, and whatever the drug we design, probably in the laboratory, in our research, we might take some 10 years to come up with a drug, okay? But then cancer cells take only overnight, okay? It will start pumping out that drug because of this enzyme, and then it will start killing the normal cells. So therefore, it's very difficult to treat the cancer cells.